Okay, now it's time to talk about meiosis. Meiosis is a process that evolved from mitosis. It is the foundational process for sexual reproduction. It's a divisional process that produces sperms and eggs, but unlike mitosis, it does not clone. Instead, it produces varieties of sperms and varieties of eggs. It's the anti-cloning cell division process. Before we dive in, you need to understand the difference between diploid and haploid states. Um, the chromosomes of most organisms are inherited from two parents. Most multicellular life on Earth is diploid, meaning that they have instructions in their cells from two different parents, uh, and so their chromosomes are in pairs. The similar chromosome pairs are called homologous pairs. For human cells, we're diploid, and in our cells we have 23 pairs of 23 homologous pairs of chromosomes. So every chromosome that mom gave you, dad gave you a chromosome that looks very much like it. It's not identical to it because mom and dad were not identical twins. Each chromosome mom gave you is part of a pair of similar chromosomes that you have in your cells because dad gave you one that's pretty much just like it except for some specific instructions. So having chromosomes in pairs makes you diploid. The diploid number for human cells is 46, and diploid is represented with the symbols 2N. Now, sperms and eggs can only have one set of chromosomes. They have to be haploid. Haploid means only one set of chromosomes. Uh, that means that a sperm and an egg can only have 23 chromosomes. And the reason for that is when a sperm unites with an egg, they will produce the magic number of 46 chromosomes in the fertilized egg. Not a chromosome more, not a chromosome less. So meiosis's job is to produce cells that have haploid sets of DNA, not diploid, and furthermore, to make a variety of different haploid cells, not clone them. This ensures the best chance for survival for offspring. Cloning is great in the short run, but terrible in the long run. Now, meiosis is very similar to mitosis, but it's two divisions rather than just one division. There's a meiosis 1 and a meiosis 2. In meiosis 1, the cell actually lines up the chromosomes, but no centromeres break, and the homologous pairs are separated. So meiosis 1's job is to separate homologous pairs from one another. In other words, separate the mom and dad sets of DNA. Meiosis 2 separates centromeres and puts individual chromatids of DNA in each of the sperm and the egg. So the two divisions of meiosis are called meiosis 1 and meiosis 2. In meiosis 1, there's a prophase 1, a prometaphase 1, a metaphase 1, an anaphase 1, a telophase 1, and a cytokinesis 1. And it's very similar to mitosis, except that in prophase 1, homologous chromosomes pair up and bind together in a process that's called synapsis. It's a sorting process. It's absolutely critical that the homologous chromosome pairs pair up. If they don't, there's no way to produce a healthy offspring. So while the homologous pairs of chromosomes are paired up, they exchange pieces of DNA. That's to maximize genetic variety in the offspring. That's called crossing over. Sections of chromosomes crisscross, simultaneously break, and swap. Now, once that's complete, then metaphase 1 starts where the chromosomes line up at the center of the cell. Then in anaphase 1, no centromeres break, but the homologous pairs separate into individual homologs, they're called, and the homologs separate from one another. And then in meiosis 2, there's a prophase 2, prometaphase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. And in this case, it's the same as mitosis, except you have half the chromosome numbers. The centromeres break, it's just like mitosis, except you have half the number of chromosomes. There is a real importance to crossing over. The exchanging of DNA between homologous chromosomes ensures that the sperms and the eggs won't be identical to each other. And this is important because even though you might be a real successful person in your generation, conditions will change in the next generation. Human technology advances. What was good for you may not be good for your kids. So you want to maximize the variety of genes that your children will get. And so that's what crossing over tries to do. You'll notice the red chromosome and the blue chromosome are homologous to each other. During synapsis of prophase 1 of meiosis 1, they pair up and then they swap pieces. So the red chromosome gets a piece of the blue chromosome and the blue chromosome gets a piece of the red chromosome. And in the end, you have 4 different sperms or eggs that are made. One's got an all red piece of DNA. Another one's got a red piece of DNA with a little bit of blue. Another one's got a blue piece of DNA with a little bit of red. And another one's got an all blue piece of DNA. All 4 of these are different, which is a good thing. It maximizes genetic variety. Now, this will summarize the phases of mitosis, but the animations are more significant, and that's what I'm going to get to next. So let's go to sperm meiosis, and we'll step through it. Here's sperm meiosis, like that would be happening in a testis. Okay, prophase 1, chromosomes form up. The centrioles separate and go to opposite sides. The spindle fibers form. But something unusual happens here before the spindle fibers show up here. The similar pairs of chromosomes find each other and lock onto each other. That's called synapsis. So this is synapsis, which allows crossing over to take place. Once the chromosomes have crossed over, the prometaphase 1 occurs where the spindle fibers attach to the centromeres. Then the chromosomes line up, but now there's no calcium release. Instead, the homologous chromosomes are separated from each other. So this is a mom chromosome, maybe that's a dad. This might be the dad, that's the mom. This is the dad, that's the mom. But they've swapped DNA, so they're no longer pure dad or pure mom. Remember, you don't make your own DNA. You got it from your parents. Every one of your chromosomes traces back to either your mom or your dad. But they scrambled your grandparents' DNA that they got so that you don't have entirely the same chromosomes your parents have. You have unique combinations of genes on your chromosomes. So now the homologs separate, and then cytokinesis 1 occurs, and then the chromosomes line up. Now you have half the number of chromosomes here. Normally, if this was a mitosis, it would be 8. This calcium release causes the centromeres to snap. The chromatids are pulled opposite directions. We have a haploid set of chromatids here, haploid set of chromatids in here, haploid set of chromatids in here, haploid set of chromatids in there. Then you end up with 4 separate sperm. They each grow a tail. Now they're ready to fertilize eggs. Egg meiosis is a bit different. Now watch this. In egg meiosis, it looks the same. Prophase, at the beginning of meiosis, the supercoiling of DNA occurs, synapsis occurs, crossing over occurs, the chromosomes line up, no centromeres break in the first meiotic division, the homologs separate from one another, and then a very unusual thing happens. A polar body forms. A very unequal cytokinesis occurs. This is because eggs need to be big and make big targets for sperm, and they have to have all the nutrients for a developing embryo for the first 6 days or so of embryonic development. So this nucleus is going to die. This one's going to live. This one over here is called a polar body. Now, the first polar body dies. Then, in a human girl's body, a female's body, this is happening before she's born. This cell then just exists in her ovary. And then once a month during the menstrual cycling, during reproductive years, this cell will get ready and it will be released at ovulation. And should it get fertilized, if a sperm hits and comes in, then the cell will complete the second meiotic division. And it will then form another polar body. This polar body would die, and then the sperm would get pulled in, and you have a fertilized egg. So meiosis in a girl actually starts before a baby girl's even 8 weeks developed as an embryo in her mother's womb. Whereas for boys, sperm production doesn't start really until puberty. So after they're born, that's a huge difference between sperm and egg formation in human males and females. So differences between human sperm and egg meiosis, here we go. Spermatogenesis begins in a human male at puberty, while oogenesis, which is egg meiosis, in a female begins at about the 7th week of embryonic development. But it stops midstream, and it leaves cells suspended in meiosis 2, prepared to do meiosis 2 should they get fertilized. So spermatogenesis starts with a diploid cell and ends up hours later with 4 sperm. Whereas in egg meiosis, the egg produces polar bodies. The polar bodies die, and the purpose is to keep one large egg with enough nutrients for the fertilized egg to survive and to be a big target for a sperm. And also it mentions here is the meiosis in a female is arrested. It's frozen between M1 and M2. They call those secondary oocytes. Then they wait for fertilization before they'll finish meiosis 2. Sperm meiosis, here it says, have equal cytokinesis. Egg meiosis has unequal cytokinesis with the polar body production. Oogenesis produces only one cell for every one egg cell that starts meiosis, or it's called an oocyte that begins meiosis to produce an egg. Only one egg gets made. Whereas with sperm, a spermatocyte that starts meiosis makes 4 sperm. So the differences here between mitosis and meiosis are laid out. One of the biggest differences, mitosis is a cloning process. Meiosis produces genetic variety. But mitosis is a single division. Meiosis is 2 consecutive divisions. Mitosis produces diploid cells from diploid cells. Meiosis produces haploid cells from diploid cells. So in human mitosis, 46 chromosomes start mitosis. Each cell has 46 chromatids at the end. In human meiosis, 46 chromosomes start out, but in the end, the sperms and the eggs have only 23 chromatids apiece. So one last big difference is in meiosis, no centromeres snap in meiosis 1. They snap in meiosis 2. And in mitosis, centromeres snap in anaphase. Now, the last thing here is just, these are mitotic cells. These are not meiotic cells. And the graphic is asking you to decide what phases the cells are in. Well, these cells that you see across the top strip, they're all in interphase. They're either in G1, S, or G2. Some of them have 2 small organelles inside the nucleus, and others only have 1. That's what produces the ribosomes. I'm having a brain cramp at the moment. I cannot remember the name of the organelle. But nevertheless, some have 2, some have 1. But these are also all in G1S or G2. They're in interphase. Now, this cell right here is in prophase. The chromosomes are starting to supercoil. These are in interphase again. Interphase, this is a prophase. Now this is an anaphase here. All these are interphase all the way across. This one may just be starting prophase. And by the way, these are the nucleoli or nucleolus. These may just be starting. It's hard to say exactly what they're doing. The fact that each of these is half-sized means that maybe they've just finished mitotic cell division. You see how this cell is larger and these 2 cells are smaller? These 2 cells may have just be starting G1 after cell division. It's hard to say, but they're interphase. This is an interphase cell here. This one right here is probably in metaphase, or it's just beginning to start anaphase. This is prophase, this is prophase, but this prophase is earlier than this one because the nucleolus here is darker than this one. And then these are all interphase again. Now that concludes my discussion on cell division. We'll talk more in class on this topic.